Cancer is the most common human genetic and epigenetic disease. The transition from a normal cell to a cancer cell is driven by the changes to a cell's DNA, which includes not only the mutations to the DNA sequences of oncogenes, but also includes the changes to DNA modifications, namely DNA methylation. Higher levels of DNA methylation have been characterized in some key genome regions in cancer cells. In our lab, we have found that the abnormal DNA methylation in cancer cells is related to another key feature of cancer cells, which is there are fewer copies of mitochondrial DNA a cell compared to a normal cell. Mitochondrion is an organelle in cytoplasm known as the powerhouse of a cell and mediates cellular activities. It hosts multiple copies of mitochondrial DNA, and the number of mitochondrial DNA is tightly regulated through mitochondrial DNA replication according to the energy requirement of each cell type. Mitochondrial DNA copy number can be as high as several thousands in cells like neurons and muscle, while it is much lower in fibroblasts, spleen cells, and stem cells. A group of master regulators of mitochondrial DNA replication, which is nuclear DNA encoded and potentially epigenetically controlled, are translocated to mitochondria to regulate mitochondrial DNA replication. My PhD project is to investigate the epigenetic control of mitochondrial DNA copy number and how this regulation contributes to tumorigenic features like cell proliferation and restrict differentiation. The outcome of my project will contribute to the understanding of tumorigenic mechanisms and also aid in the development of cancer therapies, which involves the nuclear DNA and mitochondrial DNA encoded genes. Thank you.